Pharmapal.
Inhibition of breast cancer with transdermal tamoxifen-encapsulated Lipoplex. Hello everyone. Today, I'm excited to share insights from a fascinating paper titled Inhibition of Breast Cancer with Transdermal Tamoxifen-Encapsulated Lipoplex by Lin et al. This research explores an innovative approach to breast cancer treatment using a transdermal delivery system. Breast cancer remains a significant health challenge worldwide. Traditional treatments, such as tamoxifen, can have side effects and may not be effective for all patients, particularly those with CYP2D6 polymorphisms. This study aims to enhance the efficacy of tamoxifen while minimizing adverse effects through a transdermal delivery system. So, what exactly is a transdermal delivery system? It allows medication to be absorbed through the skin, providing a sustained release of drugs directly to the affected area. This method can improve drug accumulation in the target area, which is crucial for effective treatment. The researchers developed a Lipoplex system, referred to as LPPC/TAM. Here's how it works. Lipoplex composition. It consists of cationic liposomes that encapsulate tamoxifen. Mechanism. The positive charge on these liposomes enhances their ability to penetrate the skin barrier and accumulate in tumor tissues. This innovative approach resulted in an impressive 86% inhibition of tumor growth in mice models bearing BT474 tumors. Let's dive into the experimental setup, MTT assay. To assess cell viability, the researchers used the MTT assay. Here's a quick overview. Step 1. 100 L of 2 mg per milliliter MTT reagent was added to each well. Step 2. Incubation for 5 hours at 37 degrees Celsius. Step 3. Media was aspirated, and 100 L of dimethyl sulfoxide was added. Step 4. The optical density, OD, was measured at 595 nanometers using an ELISA reader. This method allowed them to plot cell viability as a percentage of untreated controls. The results were promising. The LPPC/TAM system not only improved drug penetration but also showed significant therapeutic efficacy without damaging the skin or other organs. Key findings: enhanced skin penetration and drug accumulation, effective tumor growth inhibition, minimal side effects compared to traditional systemic treatments. In conclusion, the LPPC delivery system represents a novel transdermal tool for breast cancer therapy. It holds the potential to provide an effective treatment option, especially for patients who struggle with conventional therapies due to genetic variations. This innovative approach could pave the way for more personalized and effective cancer treatments in the future. Now, let's explore the key figures and tables presented in the paper. Each visual element is designed to enhance our understanding of the research findings. Figure 1. Study design. Description. This figure outlines the overall study design, detailing the experimental setup and methodologies employed. Key points. The transdermal delivery system is highlighted, showcasing how tamoxifen is encapsulated in lipoplexes for enhanced absorption. The timeline of experiments is clearly laid out, providing a roadmap of the research process. Figure 2. In vitro cell viability assay. Description. This figure presents the results from the MTT assay, which measures cell viability. Key points. The data demonstrates the effectiveness of the tamoxifen-encapsulated Lipoplex compared to control groups. Notice how the percentage of cell viability is plotted against varying concentrations of the treatment, illustrating a dose-dependent response. Figure 3. In vivo skin permeation. Description. Here, we see the results of the skin permeation studies conducted on nude mice. Key points. The figure shows fluorescent imaging of tumor samples, indicating how effectively the treatment penetrated the skin. The comparison between the LPPC-DI and cream-DI formulations is crucial, as it highlights the superior permeation efficiency of the Lipoplex formulation. Figure 4. Tumor growth inhibition. Description. This figure illustrates the impact of the treatment on tumor growth over time. Key points. The graph clearly shows a significant reduction in tumor size in the treatment group compared to controls, emphasizing the therapeutic potential of the transdermal delivery method. The statistical significance of the results is marked, reinforcing the reliability of the findings. Figure 5, Side Effects and Efficacy. Description: this figure discusses the side effects associated with traditional systemic tamoxifen treatment versus the new transdermal approach. Key points. The authors argue that the transdermal method may minimize systemic side effects, making it a promising alternative for patients, particularly those with CYP2D6 polymorphisms. Tables overview. Now, let's briefly touch on the tables included in the paper. Table 1, Patient Demographics. Description: this table summarizes the demographics of the study participants. Key points. It includes age, gender, and other relevant clinical characteristics, providing context for the study population. Table 2, Histopathological Analysis. Description: this table presents the findings from the histopathological evaluations. Key points. The results indicate the cellular responses to treatment, showcasing changes in tumor morphology and cellularity. In conclusion, the study by Lin et al. Offers promising insights into the use of transdermal tamoxifen-encapsulated Lipoplex for breast cancer treatment. The figures and tables effectively illustrate the research findings, highlighting both the efficacy and potential benefits of this innovative approach. 1. What are the key advantages of using a transdermal drug delivery system for tamoxifen over traditional systemic administration? Localized treatment. Transdermal systems allow for localized delivery, reducing systemic exposure and potential side effects. Sustained release. They can provide a sustained release of tamoxifen, maintaining therapeutic levels over time without the peaks and troughs associated with oral administration. Improved compliance. Patients may find transdermal patches easier to use than daily pills, potentially improving adherence to treatment. 2. How does the formulation of LPPC/TAM enhance the permeability of tamoxifen through the skin? Cationic liposomes. The positive charge on the surface of LPPC/TAM enhances its interaction with negatively charged skin components, improving skin penetration. Encapsulation. By encapsulating tamoxifen in liposomes, the formulation protects the drug from degradation and facilitates its transport through the skin barrier. In vivo efficacy. The study demonstrated that LPPC/TAM successfully penetrated the skin in animal models, leading to significant drug accumulation in subcutaneous tumors. 3. What implications do the findings regarding CYP2D6 polymorphisms have for breast cancer treatment? Personalized medicine. The study suggests that patients with CYP2D6 polymorphisms, who may not metabolize tamoxifen effectively, could benefit from transdermal delivery, providing an alternative route of administration that bypasses metabolic pathways. Reduced side effects. This approach may minimize the side effects associated with systemic treatment, offering a tailored therapeutic option that maximizes efficacy while reducing adverse reactions. 4. What limitations were identified in the study regarding the transdermal delivery of tamoxifen? Skin permeation. Despite the success noted, the study acknowledges that skin permeation remains a major limitation for many drugs, including tamoxifen. Animal model. The results were obtained from animal studies, which may not fully translate to human physiology, necessitating further clinical trials to validate efficacy and safety. Long-term effects. The long-term effects and potential toxicity of sustained transdermal delivery need to be investigated to ensure patient safety. 5. How do the authors suggest future research should proceed based on their findings? Clinical trials. The authors recommend conducting clinical trials to evaluate the safety and efficacy of LPPC/TAM in human subjects. Formulation optimization. Future studies could focus on optimizing the formulation to enhance drug delivery and minimize any potential side effects. Broader applications. Investigating the use of similar transdermal systems for other therapeutic agents in breast cancer or different types of cancers could broaden the impact of this research. Thank you for joining us today. We hope you found this video informative and interesting. Don't forget to like and subscribe to our channel for more interesting content like this. Thanks for watching.
Pharmapal.